Yalla girls, let's start a new lesson today. Um, it's page 377. Get your books to follow with me. Okay, it is about gene regulation and expression. In the last lessons, you remember that we have discussed and learned about genes and how genes are transcripted and how genes at the end can form a protein. Gene is all about our gene expression. It is the thing to do. Girls, please mute your mics, please. So gene expression, it means the genes forms, some steps are formed so that the codes or the information on the gene is expressed into a certain protein type. If we make some quick revision here, you remember that a segment of DNA is changed into messenger RNA by what? By a process called what? Huh? A segment of DNA is coded or copied into a messenger RNA by a process we call it what? Yalla girls, do you remember? Yes, transcription. Transcription. Very bad handwriting, okay? But you understand me, of course. Transcription, okay? Type. From messenger RNA, okay, to transfer RNA. Okay, from messenger RNA to transfer RNA. This what we call it what? The information coded on the messenger RNA is done with a process by tRNA to change it to the last two proteins. Okay, this process is called what? The messenger RNA to tRNA and protein. This process is called what? Tell me. So transcription and then the code in the messenger RNA is translated by the transfer RNA into a protein. Hmm. This process is called what? Mute your mics, girls. Just type translation. It is translation. You remember? So a segment of DNA will be changed into a messenger RNA by transcription, and this messenger RNA goes into the cytoplasm to be translated, especially in the ribosome, by the transfer RNA into different types of amino acids assembled together by the ribosomal RNA into a protein. Okay, can you hear me? Okay. Now let's proceed here. Let's listen first this part and think about what do you hear? Yeah.
Think about it. Think of a library filled with how-to books. Would you ever need to use all of those books at the same time? Of course not. If you wanted to know how to fix a leaky faucet, you'd open a book about plumbing, but would ignore the one on carpentry. Now picture a tiny bacterium like E. coli, which contains more than 4,000 genes. Most of its genes code for proteins that do everything from building cell walls to breaking down food. Do you think E. coli uses all 4,000 plus volumes in its genetic library at the same time?
Okay, girls, what about the idea here? What about the idea here? I want someone to open her mic and tell me what is the idea here. What is the idea about the genes and the whole genes? What to think about here? Trying to trying to get you to think about. Huh? Okay, Lujain. Hello, Lujain. Okay, girls, any one of you had heard think about it. What is the idea in think about it? Huh? Hagar, can you hear me? Hello, girls, can you hear me? Who can hear me? The question is, think about it. This part that we have read here, or that you have heard here, okay, okay, what is telling you about? What's it's telling you to think about? Hagar, يلا يا Hagar, open your mic and tell me. What is the idea here, girls? Just I want to give you an introduction about the idea of the lesson. طيب, I will ask you questions. If we have, for example, this Escherichia coli or this bacteria here, it tells you to have like around 4,000 genes inside it. 4,000 genes inside it. Do you expect that all these genes would work in the same time or would work always? Would you expect that? Hello, babies. Okay, sorry, Hagar, but I couldn't hear you. But can you hear me now? Girls, can you hear me now? Okay, Hagar can. Who else? يلا يا سمارة. طيب, I'm asking you here a question, girls. Uh, okay, before. Who is Ghada Muhammad? Someone came into our meeting that is not in our... Okay, it's okay. It's okay. Girls, so could you expect that all 4,000 genes in the E. coli bacteria to work all the time? All the time they are working? Do you expect that? Okay. The Basant and Sama, they say yes. Hagar say yes. The 4,000 genes all the time, 24 hours, they are working. Okay. What? Stop. I'm asking you another question. What if some genes in the E. coli bacteria, for example, some genes do not want to make their proteins or their specified proteins by now? The bacteria does not need any proteins for some genes now. Would they even work all the time, wasting energy, wasting time, wasting enzymes? Okay, so the answer it is no. Genes cannot is not working all the time. Genes also only works or put into action when the specific protein or its specific proteins are needed. Whenever a protein is needed, its gene, like it is put on to work to get this protein. But whenever we don't need this protein, what's the role and what's the idea of working and wasting time and wasting energy to make unneeded proteins? Okay, let's return back. So how genes are regulated, how genes said to work at some times and to stop working at some times. So to conserve energy and resources, prokaryotes regulate their activities using only those genes necessary for the cell to function. Whenever the cell wants to function or want to produce a certain protein, genes work, or the specified genes only works. Okay, so how these are regulated, how genes are turned on and off. Genes are turned on and off. How genes are turned on and off? By a type of binding proteins, by proteins that regulate the genes by controlling transcription. What is transcription? As we have said just before, it is when a segment of DNA is copied or transcribed into a messenger RNA. This process is controlled by some proteins in prokaryotes. Some proteins bind to the DNA and stop the process of transcription. Okay, they switch genes on, and while others turn genes off. Okay, type. What are these type of proteins that switch on and switch off the genes? They called operon. Operon. It is a group. Sorry, girls, just a minute. So how to turn gene on or off? By binding proteins. What is the meaning of operon? Operon, it is a group of genes that are regulated together. It means that genes that in an operon usually have related function. What does that mean? To make it more clarifying here. Let's move to the next page. Okay, girls, here there are three genes. There are three genes that they work together, or they work to form proteins that have the same or relating function. Here we have three genes. Okay, we have three genes that when they are switched on, they form enzymes that are functionally related together, or they do are relating functions together. Yani genes that work in a certain function, one function together. They are, when they are accumulated or collected together, we call them operon. We call them operon. Let's return back to the definition. So operon, it is a group of genes, okay? A group of genes that are regulated together. They are found beside each other and regulated together to be easy, or to be, yani, like to facilitate for the cell whenever a certain function is needed with a certain types of genes for this function to be found together, to be found next to each other. The genes in an operon usually have related functions. What makes the genes coming together or coming after each other in a part of the DNA, that they have related functions. Again, what is an operon? It is a group of genes that are regulated together to form a related function. Is it understood? I want you to operon to understood it fully. Is it understood? Girls, hello. Okay. Type. I will repeat again what's the meaning of operon. Okay. Type. Here, let's move to the figure here. So it will be, you see here, this is a segment of DNA. This is a segment of DNA. I just only want you to, yeah, just to concentrate here. These are genes. These three things here together, or aside to each other, they are three genes. They are three genes. We call them lac genes. Lac, it refers to lactose, to the sugar lactose. Lac refers to the sugar lactose. So these three genes, they regulate the lactose sugar, or the fermentation or the process of lactose sugar inside the E. coli. So these three genes, they are having a related function. What are the related functions? That they control the fermentation or the process of the lactose sugar inside the Escherichia coli. Okay, when I'm saying operon, it is when we find a group of genes regulated together aside to each other, where they are forming a specific or a related function. Because here these genes, they all have to play a role in the lactose fermentation. They are put aside to each other or regulated together as one unit. Okay, we call this an operon. We call this an operon. Huh? Is it understood? Yeah, all of you, if you want me to turn back, it's okay. To tell it again, it's okay with me. Don't go to the next point without understanding the former one. Huh? Type. Okay. So an operon, it is a type of or group of genes that regulated together. Okay, they will be regulated together because they have the same function, or they have related functions, and you will find them assigned to each other. Type. As an example to explain the prokaryotic gene regulation, we are going to speak about the operon, or a cluster of 3 genes that must be turned on together and turned off together before the bacterium can use the sugar lactose. Lac operon, it is denotes the lactose. Lactose, the Escherichia coli, it is a type of sugar that the Escherichia coli feed on. If the lactose is found around the bacteria or in its environment, it has to use it. It has to make it enter the cell membrane. It has, after it enters the cell membrane, it have like to ferment it. It means to separate, because the lactose have inside it galactose and glucose. It has to separate between the galactose and glucose to use the glucose at the end. So some things have to be done whenever lactose is found in the environment. How Escherichia coli or E. coli manage to do that? By some proteins, by the formation of some proteins. How these proteins, from where these proteins come? It comes from genes, from coding genes, by the effect of transcripting of 3 genes. We call them the lac operon. These 3 genes, or the lac operon, control the formation of proteins needed for this process of feeding on lactose. Is it understood? Okay, which part do you want me to repeat? From where? The operon or the last part I said? Type. For the operon, did you understand the term of what's meaning of operon? Type, the last part. Type. Let's go to our board here. Type. If I say, for example, this is the Escherichia coli, okay? This is the Escherichia coli. This is the bacteria. Okay. Escherichia coli feeds on lactose. Lactose sugar, if you remember, we have taken about the lactose sugar. It is a disaccharide that consists from glucose, okay, plus galactose. Okay, Escherichia coli feeds on this type of sugar. Okay, whenever lactose is found, Escherichia coli wants it to enter inside the cell, and inside the cell, it wants to break the bond between the glucose and the galactose, so that it will use the glucose as a food. Okay, how lactose enters the Escherichia coli cell, and how inside the cell the galactose and the galactose are being separated. Escherichia coli uses what? Enzymes to do so. It uses enzymes to do all these functions. What are enzymes? At first, what is the nature of enzymes? They are proteins. So in order for the E. coli to do this process of feeding, it needs a certain proteins. Okay, how proteins are assembled? What controls proteins to be formed? Tell me, what tells or what specifies certain proteins to be formed? Yalla, answer this question. What initiates from the nucleus the process of forming proteins? What tells this protein to be done and this protein not to be done? Tell me. What makes protein to be done? What in the nucleus, Yasara? What is a nucleus that controls the formation of proteins? What in nucleus? Which part of the nucleus which manages or controls the proteins done or proteins assembly? Hi, Sumaya. Yeah, girls. No ribosomes. Ribosomes, it is the site where proteins form. But what controls the formation of proteins? The control. Ah, not asking about the site. Instructions from where? Yes, instructions from. Yes, structure from genes. Genes. So genes give instructions for proteins to be formed. Yes, so if I return back here to the enzymes, which are proteins, how they are formed by structures from genes. So خلينا يعني ثابتين في النقطة دي genes. Let's go back to our book. Okay, so genes. Okay, here these genes, okay, these lag genes, they are responsible, or they carry the instruction for the formation of the enzymes needed to form or to form the process of lactose feeding, as we seen in the board. Is it understood? Hmm. Let's proceed, and inshallah you will like understand more. So this part, it is the operon. Okay, these are the lag genes, three genes that have the instruction formation of certain enzymes that is needed by the Escherichia coli to ferment and enter the lactose inside it. All the time Escherichia coli wanted these genes to process proteins, the answer, or process enzymes or the lactose enzymes, the answer it is no, because not all the time it wants to feed, not all the time the lactose is found around it in the environment. Maybe sometimes it feeds on another materials. So the Escherichia coli, question here, do the Escherichia coli needs these lactose genes to be switched on all the time? Yes or no? Do the E. coli needs these lactose genes or this lactone operon to be switched on all the time? Yes or no? The answer is no. This gene is not needed all the time to be turned on, because not all the time the bacteria maybe it is feeding on another type of lactose. Maybe lactose is not found. Maybe it's not hungry anymore. Okay, so what controls the switching on and switching off of this operon or of these genes, as we have خلاص يعني اتفقنا ان هم they will not be switched on all the time. طيب, we have here an oocyte and P site. Oocyte before these lactose genes or lactose operons, we have oocyte and P site. The P site, it is the site where the RNA polymerase binds. The P site or the promoter, if you remember, it is the site where the RNA polymerase binds. You remember what is the function of RNA polymerase? Yalla quickly. What is the function of the RNA polymerase? It forms what? Huh? In the process of what? The first process of protein assembly, it is what? The first process خالص in the nucleus for the assembly or the protein synthesis, it is what? When a segment of DNA, it acts as a paste or to copy a segment of messenger RNA, and this is called, we call it what process? When a segment of DNA acts as a part or a copy to copy, and act as a segment to take a copy from it on messenger RNA. We call this process what? Is it transcription, duplication, or translation? Transcription, duplication, or translation? Hello. Yes, it's transcription. Why, girls? You didn't study these lessons? It is one of the basics, يعني, to know the transcription. It means that a segment of the DNA is used to make a fragment of messenger RNA carry its instruction for formation of some proteins. This is transcription. Yes, it is transcription. So let's return back. Okay, you will have a quiz on inshallah next Sunday on chapter 13, yeah, girls. So start now. يعني started to study. Okay, so the RNA polymerase, it binds here to start the process of transcription of these genes whenever it is needed. But guess what? Who is standing here in the middle? What in the O side? It is an operator. An operator, it is a type of protein that represses the process of transcription of these genes. Again, B, it is the site where RNA polymerase binds here to start the transcription of these lag genes whenever needed. Who is in the middle here? This is a protein called an operator, a repressor, binds here to like stop or like to, like it's acting like a soldier, stopping the RNA polymerase to start transcripting these genes because RNA polymerase has to be in direct contact كده يعني very aside to the lag genes to start transcripting them. Whenever the O or the binding protein or the repressor protein binds here, يعني it's like it discriminates between the RNA polymerase and the genes, so the process of transcription will be stopped as long as the repressor protein is binding here. It discriminates between the promoter or the RNA polymerase and the lag genes and stops the process of transcription. Okay, here comes if the operator here or the repressor, this is the repressor protein is binding here. Whenever RNA polymerase comes to bind here, it cannot be. Okay, when the lactose is not present, the repressor protein binds to the operating region here. This blocks RNA polymerase will be blocked and it cannot transcribe the lag genes. Okay, this is the RNA polymerase coming want here to bind to this side of the P site, but it cannot because here we have the repressor protein binding to the O site. Till now, is it understood? Okay, who else? Is it understood? What happens if the lactose now is present and the cells need this lag operon or lag genes to be switched on? Okay, what happens? Here, this green one, they are the lactose. They are found now. And so the cell now, or the Escherichia coli wants these lag operons to be switched on. What about the operon that was stopping here the process of transcripting the transcription or of the coding of the messenger RNA, of the genes? The lactose come to bind with this the lactose come to bind with this repressor protein, changing its shape. Like as you see, when changing its shape, it's like released from the site, where it was standing here to stop RNA polymerase. When it is detached here down from the segment of DNA polymerase or RNA polymerase, could find the way opened to start to the form of transcription of messenger RNA to switch on these genes or the action of these genes. Huh? What do you think? Huh? Is it clear? يا حلوين. Is it clear?